Welcome everybody, and I'm delighted to introduce our next speaker. So today we're hearing from Professor Ara Monadjem, based at the University of Eswatini, formerly known as Swaziland. And Ara is going to be talking to us about taxonomy and how it can assist in the conservation of African plants. Ara is a well-known researcher and he's traveled extensively across Africa investigating taxonomy, diversity, and also distribution. And I'm delighted to say that the second edition of the Bats of Southern and Central Africa, which Ara is a co-author, along with Peter Taylor, Woody Cottrell, and Corey Schoeman, is going to be coming out in August this year. So we're all really looking forward to that. Ara also sits on the IUCN Bat Specialist Group. He's our African lead for that. He's also an advisor for Bat Conservation Africa and was instrumental in setting it up, and also Bats Without Borders. Also supported a lot of African students, masters, undergrads, and PhDs, including myself, but also international students researching bats in Africa. Thank you very much, Ara, for your time, and over to you.
Hi, good afternoon, evening, morning to all of you, and thank you for joining this, my very first webinar, coming from someone like me who absolutely hates technology for human interactions. This is a bit difficult, and well, we'll see how it goes. So before I start my talk, let me just say that this is a talk that is based on my presentation last year at a plenary session at the African Small Mammal Symposium. So if any of you were there, I apologize. You probably will have heard at least half of this talk already. The theme of the talk is, well, to give you some background, I'm an ecologist, or at least I'm supposed to be an ecologist, and most of my working life I've worked as an ecologist, and I was always a bit skeptical about taxonomy and systematics, and thought it was something we should leave to other people to do. What I want to try and show you in this presentation is that that is a completely wrong attitude and approach, and that in fact taxonomy and systematics are very integral to all the disciplines of biology, especially ecology and conservation. And I will draw mostly on my own experience with this through the work that I've done on bats in different parts of Africa. So there we go. So bats, as everyone on this group is already familiar with, bats are absolutely fantastic, wonderful creatures, very diverse, with different life histories, different morphologies, and all of them very cute and beautiful. This is just some photographs I've taken of some African bats, and we are all aware that bats are very species rich. And in fact, if we look at Nancy Simmons's website, her latest count as of a few minutes ago was 1,421 species. Familiar with the site, please do visit it. It's very useful for getting the latest information on number of bat species, names, taxonomy, and so on. www.batnames.org. So bats are diverse, but how diverse are they compared to other mammals? I'm sure, again, most of you are bat people, you're very familiar with this. There are approximately 6,500 species of mammals, at least according to the Burgeon database. And about 40% of those species are rodents, and 20% are bats. And this is what this diagram is showing. A large chunk of the mammalian diversity comprises bats. If we turn to Africa, and if we look at what the situation is on this continent, And if we use the resource, the beautifully illustrated and written Mammals of Africa by Kingdon et al., then you'll see that the same pattern holds for Africa, approximately 460 species of rodents, and about half that number of bats on the continent. And the iconic African mammals, the antelopes and the ungulates, primates, carnivores, in those yellow bars, you can see that they hold far fewer species than the little mammals. Now, if we look at how much research has been conducted on these groups, we see the exact opposite trend. The majority of papers are written on the larger mammals, typically for 3,000 plus papers if you use the Kingdon Mammals of Africa volumes, 3,000 plus citations for primates, similar numbers for ungulates, and 2,500 for carnivores, but half that number for rodents and even less for bats and shrews. But this doesn't really tell the full story because there are many more species of rodents and bats. So if we divide by number of species, this is what the graph looks like. This tells us how many papers have been published per species on average. And now you can see that for the large mammals in Africa, somewhere between 30 and 35 papers per species. For rodents, it's around 7 papers, so a fifth of the amount. And for bats and shrews, it's only 4 papers per species. So we have a real big distinction in research effort, or division in research effort, between the large iconic mammals and the smaller but far more diverse groups. And one of the problems with studying small mammals, and particularly bats, is that there are problems with taxonomy. There's problems with identification. We don't have good keys. We cannot identify the bats very easily in the field. Some of them, this is because we don't know enough. Others, it's because they're truly cryptic and share similar morphologies with other similar species. There are lots of bats that go around with no names. Here's 2 examples, or one example of 2 African bats that are widespread and common. These are what we call Hipposiderus cf. ruber. It is not Hipposiderus ruber, but it's 2 species that are widespread in West Africa, but neither of them has a name. Neither of them can be identified in the field either. They look identical with similar body measurements and similar leaf noses. The differences in the photos here are not because they're not real differences. This is just the way that the bat is looking at you at the moment. The only way we can tell these 2 apart is through genetic analyses or with a bat detector. So this makes it a lot more challenging to do ecological research on African bats. And as a result, possibly we have fewer studies on these groups. So if we're going to do any good conservation, good effective conservation, we need to know what the species are that we want to conserve. And this is where taxonomy becomes very important. And we have had a helping hand in the last few decades, thanks to genetics, particularly molecular biology and sequencing. And I'm not going to go into any details here, just to say that evolution is a pretty complex process. And as a result, we can end up having species that are morphologically similar, but that may not necessarily be closely related. This happens due to convergence. And then sometimes we've got 2 species that don't look alike at all, or are ecologically very different, but in fact are very closely related evolutionarily. And genetics has allowed us to get a handle on this. The reason I bring this up is that occasionally we are still doing taxonomy without reference to genetics. And here's one example of that. It's in fact a very good paper. I don't mean to belittle this paper at all, but it's just a useful example of how we can arrive at different conclusions with and without genetics. So this is a paper published by Deanne Reeder and her colleagues in 2013. And what they proposed was that this species that was previously called Glauconycterus superba, and you can see why it is called superba. It is an absolutely beautiful bat, stunning, possibly the most beautiful bat in Africa. They, through very careful morphological analysis, came to the conclusion that this species is distinct from other species of Glauconycterus, and it's distinct enough that it should be placed in its own genus, Nyumbaha, that they coined. But this was based purely on morphological characters, no genetics at all. At the same time, or slightly later, Alexander Hassanin from France, together with a large group of co-authors, was working on this genus. This is the genus Glauconycterus. This is an ordination plot. I don't want you to worry about the details here. I want you to know that each different color represents a different species of Glauconycterus. So for example, up here, I don't know if you can see my cursor, but the dots up here, that's Glauconycterus variegata, which is lying close to the blue Glauconycterus argentata, showing that these two species are similar morphologically. And you can see that most of the Glauconycterus is sitting in the band to the left and to the top of the ordination. However, in the purple circle, you can see the Glauconycterus superba or Nyumbaha superba. And what you will notice is that it is distant from any other Glauconycterus. In other words, this is showing, it's supporting what Deanne Reeder and them found in the morphological analysis. This is a very distinct species morphologically from other Glauconycterus. However, when we look at the genetics, we get a very different story. Here, the details are totally not important. All I want you to notice is that in the red box that I've highlighted here, that is where Glauconycterus superba lies. And what I want you to notice is that it is embedded within the tree of Glauconycterus. In other words, genetically, Glauconycterus superba is not any different from other, is not majorly different from other members of this genus. And therefore, it should not be in its own genus, or if it is moved to a different genus, it will require other species within Glauconycterus to move with it. So this is just a little aside to show how important genetics is. And I will be reverting back to genetics and phylogenetic trees for much of the first half of this talk. And I apologize if that is going to get a bit heavy and a bit boring for some of you. What I'd like to do next is to move on to a rather difficult group of bats, difficult from an identification and taxonomy point of view. The LBJs of the bat world, the little brown jobs, as the birders call them, difficult to identify and therefore typically not identified and left to others to deal with. So these beautiful little bats, as similar as they may look physically, they all have very interesting stories to tell. And I would like to look at one group of these vespers, the African pipistrelloids, or the pipistrelle-like bats, which have had a very checkered history with splits and lumps and shifts and further splits and further lumps, so much so that it is confusing even for people that are working on the group on a regular basis. So if we look in Mammals of Africa, the same volume that I mentioned a bit earlier, if we will find that all these pipistrelles are lumped into one genus called Pipistrellus, with a few larger ones pulled out into Eptesicus. But even before Mammals of Africa was published, for example, in Simmons 2005, and since Mammals of Africa as well, these pipistrelles are typically placed into three genera: Hypsugo, Pipistrellus, and Neromisia, at least within the African continent. But how these genera relate to each other and how they relate within themselves is really very poorly understood. And I would like to illustrate this by the work that I've done in West Africa. So I'm going to show a series of phylogenies, phylogenetic trees. And again, the emphasis is not on the detail. What I want you to take away is the broad message that there is a lot of confusion in these groups. So this is work I did in 2013, based in Mount Nimba in Liberia, and you can see that there are three main groups within the tree, which I've labeled Neromisia, Hypsugo, and Pipistrellus. But can you see that Neromisia is in fact paraphyletic? It appears in two different branches of the tree. The two Neromisia's groups don't appear to be directly related to each other. So this is obviously not good from a systematic point of view. You cannot have two species in the same genus in this manner. But then a short while later, together with Jan Decher, we did another revised phylogeny for bats of the same region, but this time combining data from Guinea, where Jan Decher was working. And this time we were able to recover Neromisia as a monophyletic group. So Neromisia, Hypsugo, and Pipistrellus all seeming to behave very well. And then 3 years later, on an even bigger data set, again from the same region, Liberia, Guinea region, the forested parts of Liberia and Guinea. And again, you can see that Neromisia is giving a biologists in particular get very frustrated. They say, why do you keep changing the names? It goes from this to that. Just as we learn this name, we have to learn a new name. So this is just as a way to explain why these names keep changing, because taxonomists are trying to make sense of the relationships between these groups. One other thing important thing that came out of this paper that was that led by Rainer Hutterer, that is a colleague of Jan Decher in Bonn in Germany, is that in fact the genus Hypsugo, the African members of the genus Hypsugo are not closely related to Hypsugo itself, and we named these a new genus Parahypsugo. And you can see from this tree that Parahypsugo is equally related to Hypsugo as it is to Pipistrellus, which was the basis for us recognizing it as a distinct species. There's also morphological differences. Just one morphological difference that I want to point out, a very obvious one in Hypsugo. This is very obvious if you have the skull in your hand. It's not obvious when you're looking at an image on a screen like this. But if you look at a Hypsugo skull, the rostrum, the snout, has two deep pits, one on either side. Very, very obvious. But in Parahypsugo, that rostrum is smooth. Those pits are absent. So it's not just genetics, there are also morphological characters that support this genus. However, and here we come back to the same problem of paraphyly. The genus Parahypsugo is not monophyletic. And I've named the different genera here. I've color-coded them with different colors. Parahypsugo is the straw-colored brown. Pipistrelle is the violet, and Nycticenops is the red. You can see that Parahypsugo sandwiches around Pipistrelle grandidieri, and therefore either Pipistrelle grandidieri is a Parahypsugo, or in fact what the case is that Parahypsugo has an older name, which is the subgenus of Pipistrelle hypsugo, which is Afropipistrelle. So I don't want to get in the details here. All I'm trying to show at this stage is that taxonomy can be very confusing to an ecologist, because all that the ecologist wants is a name. Give me the name of this animal, but giving a name is not as easy as you might think, because of the problems that I've tried to illustrate over here. And that is not the end of the story anyway. I managed to spend a few very fruitful months in the lab of Bruce Patterson at the Field Museum, and we have worked on revising the entire African Pipistrelle-like bat group, and that manuscript is going to be submitted this week or maybe next week. Because of, well, I cannot go into any details at all, but I will just ask you, please be on the lookout for it, because I think for the first time we're going to bring some stability to this group, and I think from now on, the way we have defined the groups, I think from now on we can have a stable base on which new species can get names within this group. So I will leave it. Sorry to leave it hanging like this, but in a few months' time, hopefully all will be revealed. So now that I've tortured you with talk of phylogenetic trees and with taxonomy and so on, systematics, I want to put that aside, and I want to move on to illustrating how taxonomy can help conservation. And I'm going to give several examples, 3 examples to be exact, of cases where I have the work I've done personally in taxonomy has helped with conservation. The first example comes from West Africa, from Mount Nimba, and this is the case of Hipposideros lamotti, which is and this is work that was done in collaboration with Aisling Dawa, who works for Golda International. They were subcontracted by the mining company to do an EIA, and one of the things that came up was Hipposideros lamotti, which was described relatively recently by André Brosset in 1984 from Mount Nimba, and reputedly an endemic to Mount Nimba itself. So Aisling was having problems identifying whether the bats she had caught were Hipposideros laboti or not. So she called me in to come and give a hand. And just to put Mount Nimba on the map for you, because I'm going to talk about Mount Nimba several times in this talk. This is a map showing species richness in Africa, species richness of bats. The brown colors are the highest diversity. The dark blue colors are the lowest diversity. This is a map based on work that I published in 2018. But in fact, if you want a high resolution, a better high resolution map, please look at Herkert's 2016 map that he published in Ecological Modeling. So Mount Nimba is situated in a West African hotspot. And it is absolutely gorgeous location. You can't see very clearly on this picture because it is never clear. In Mount Nimba, there's always haze in the air, or it is raining and cloudy. But it is a mountain that rises up to 1,700 meters, the base of which is tropical rainforest. And as you move up the slopes of the mountain, or the edaphic savanna, the savanna that is maintained through fire. So I had been working on Mount Nimba for many years before Aisling called on me. And based on the work I'd done on the Liberian side of the mountain, I could tell her that I have 6 species of Hipposideros. At that time, Cyclops and Gigas were still under the genus Hipposideros. Each blue bar in this phylogeny here represents a different species. Dorirhina cyclops at the bottom, Macronictrus gigas, and then Hipposideros marusi, beatus, and then the 2 Hipposideros that I showed you earlier on that don't have names, that in fact, they do have names. They're called lineage E and lineage C. So when I took the tissue from the Lamoti, putative Lamoti, it came up with a blue bar with a question mark. So the question really was, is this bat from Mount Nimba really Hipposideros lamoti or not? This is a photograph of that bat. If you look at the echolocation call of lamoti, it's distinct, calls at about 118 kilohertz, very distinct from any of the other Hipposideros calling in that region. For example, the common Hipposideros CF ruber calls at about 135 kilohertz. But the problem here is that the measurements of lamoti that we got were not consistent with lamoti. So if you look at the measurements of this bat as they were taken by André Procé and under the holotype, he measured 55.6 for the forearm. The mean forearm of my 6 bats from Mount Nimba was 55.9. There's no problem over there. Skull length, there's no problem. Zygomatic arch, not really a problem. Mandible, 3rd metacarpal, no problem. The problem is when you look at the first phalanx and the second phalanx. There is a massive difference between the measurements of André Procé and those measurements that I took in the field. I took of specimens that we captured in the field. And the problem here is that the Hipposideros lamoti has traditionally been identified based on its very shortened first and second phalanges. So the only option I had was to look at the type specimen, which is at the Paris Museum. I was working with Christian Denis on the Rodents of Africa book at the time. And in 5 Rue Buffon. Anyone who's been to Paris will know that the museum itself is beautiful, but where the mammal collection is kept, 5 Rue Buffon looks like something that has just managed to survive a war. Its walls are, from the outside anyway, they are quite shocking. Everything looks run down and falling apart, but in fact, it is not at all like that once you're inside. So this was the specimen. This is the type specimen of Hipposideros lamotti. And then I remeasured that type specimen, and lo and behold, if you look at the first phalanx or the second phalanx, my remeasurements of the type are in brackets, and you can see that my measurements are very similar to that of Posey, but except for the first and second phalanx, my measurements are the same as mine from the field. So the conclusion here is that yes, those specimens from Mount Nimba, the top of Mount Nimba, are indeed Hipposideros lamotti, and we were able to redefine lamotti based on this new information that came from these important collections. So before I get onto the next two examples, I want to quickly also illustrate how important basic survey work is in Africa because of the rate at which we are describing new species. This is a very interesting paper that was published a couple of years ago, but predicting where the how many species still need to be described, or how many species have not yet been discovered on the different continents. And within the African continent, or more precisely within the Afrotropics, Fisher estimated that there were 122 undiscovered mammals, which was the highest for any of the regions in the world. The next highest was the Neotropics. I don't remember the figure. I think it was 97 or somewhere around there. But whatever it was less than what it is for Africa. And this figure might look big and robust, but in fact, most people, when I told Julian Kerbis Peterhans this figure, he laughed, and I think he laughed because he knows that there are probably that many undescribed rodents in Africa alone, let alone all the other creatures. To show you the reality of that prediction, I'll go back to Mount Nimba and use it as an example of how basic survey work has uncovered a whole host of new taxa. This is a phylogenetic tree. The details again are completely unimportant. All I want you to note is each blue bar represents a different species. This is the Vesper-like bats, the Pipistrelle-like bats in Mount Nimba, so the Neremiscias, the Pipistrelles, and the Hypsugos. And if you count those up, you'll see that there are 10 blue bars. But of those 10, 4 of them were new to science that we described. Neremiscia rosefieri, we described in 2013. Neremiscia isabella, we described in 2015. Hypsugo hapoldorum, which is of course Parahypsugo hapoldorum, we described in 2018. And the fourth one, it's been accepted and should pretty soon, I hope. It's a new species of Pipistrelle. So, just to summarize, out of 10 Pipistrelle species on Mount Nimba, 4 of them were completely new to science. These were not split subspecies or taxa that were known and we now just formally put names on them. These, in every one of these cases, these were brand new original discoveries. And what I always enjoy showing is this, this quotation from André Brosset's original paper where he describes Hipposidrus lamotti. The team of Patterson and Demos and the crew from the Field Museum in Chicago, they've really done a fantastic job of showing us just how much diversity there is in the bats of Africa that still needs researching. And I was going to give a very short example. I'll make it even shorter now. One example is the genus Rhinolophus that they published on recently. And what I wanted to show you was, again, not the details. The details are totally not important here. What I wanted to show you rather is just how confusing the situation is and how much more diversity there is than we think there is. So if you look at the phylogeny on the left, the one that says nuclear, and you look down to where it says clivosis, the clivosis group down here, it's actually the Pheromecrium group, but you can see there's clivosis 1, 2, 3, 4, 5, and 6. So what we are calling one species, Rhinolophus clivosis, is multiple different species. If you look higher up in the top of the branches here, there's the Fumigatus eloquens group. 1, 2, 3, 4, 5 different distinct lineages that represent that, and on and on and on. So to think that we are anywhere near close to understanding the true diversity of bats in Africa, we are just kidding ourselves, completely fooling ourselves. So the third example, a very short example of how taxonomy and basic survey work can be important to conservation. Here is the distribution of a species that we described in 2013, Miniopterus mozambicus. It occurs throughout much of Mozambique, in Malawi, some of Zambia, and recently the Patterson group has also recorded it from East Africa. So it's widespread in the region and nothing really to worry about. However, in a recent publication that has just been accepted, we have shown that the lowland species is a different species from the highland species. They're completely distinct. You can even distinguish them morphologically. That is how different they are. And this new species occurs only on the top of mountains in this region, and has a very, what we are predicting, a very narrow range, distributional range. It's associated with the highlands of northern Mozambique and southern Malawi. And I've just put in a few of the mountain names there for you to see. So far, we know that this bat occurs on Mount Gorongosa and Mount Nomuli and Mulanje, but it probably occurs throughout all of those mountains. All of these mountains are under severe pressure, and they are being deforested and are likely to disappear very quickly. So here's a species that seems to be entirely restricted to these very endangered forests, and further highlighting the importance of this region. Please look at the work by Julian Bayliss. There's a photograph of him. And if you're interested, have a look at that YouTube video. It's called The Lost Forest. It's a National Geographic video that highlights work we did on Mount Liko, one of the mountains over there. So the last topic, or the last example, is it takes me back to West Africa, a region that I really love very much. And working in the tropical rainforest zone of Africa, which extends from the Congo Basin, as we know, through the lower Guinea rainforest and into the upper Guinea rainforest, which is what occurs in West Africa. So I'm going to focus on the upper Guinea forest. It's an extensive forest, or I should say it used to be an extensive forest, stretching from Togo in the east all the way to Sierra Leone in the west, approximately 1,600 kilometers. But it's been heavily fragmented, and only a few patches of it remain, except in Liberia, where there are still large chunks of forest. What I want to do, very small part of the Upper Guinea rainforest zone, and that's the borderland region between Liberia and Guinea. And this is an area of upland habitat, areas going above 1,000 meters. It's very rare in this in the Upper Guinea rainforest system. Most of the Upper Guinea rainforest is forest at 500 meters or below above sea level, whereas in this upland region, many of the peaks here are well over 1,000 meters. And in contrast to the Upper Guinea region, this upland region is a very small area. At the very most, it stretches 220 kilometers from north to south. And some of the better known ranges within here, the Mount Nimba, the Simandar range, Ziama, Wanagizi, and Walagizi. I've surveyed 4 of those 5 areas, and together with Jan effective comprehensive list of the bats for the region. So here's a table showing the number of bat species that occur in the Upper Guinea forest and the number that are endemic to that region. So there's 90 species known for the Upper Guinea, of which 12 are endemic. If we look at any one of those upland areas, and I'm choosing Mount Nimba because it has the highest diversity, we will see that it doesn't really compare very favorably with the Upper Guinea in total. At Mount Nimba, there are no endemics at the other mountains, and there's only 59 or 60 species compared to 90 species in the Upper Guinea. But if we group the Upper Guinea zone together, we get a very different pattern. If we group that those upland areas, there's almost 70 species of bats out of the 90 that occur in the Upper Guinea region, and more importantly, 9 out of the 12 Upper Guinea endemics are in fact endemic to that strict upland zone of that I showed earlier. So that upland area, which doesn't even have a name because it is not recognized as an important area, that upland area is a very important area for the conservation of African bats. Here's a list of endemics to that region. And what I want you to notice is that many of these bats, in fact, have been described in the last 20 years, since 2000 onwards, 2002 with Rhinolophus ziama. So a lot of this work has been based on very recent work. So I hope that I've managed to convince you that taxonomy, quite the opposite of wanting, taxonomists do not want to make the life of ecologists miserable. Relies on a good museum base with specimens and with molecular biologists that are going to team up with the traditional museum biologist to be able to make some sense of this massive diversity. Africa is lagging far behind with the we need to give taxonomists a little bit of slack and allow them to be able to do this very important work. So just to end off, I'd like to say, please look out for the updated bats of Southern and Central Africa with an updated taxonomy and systematics for all the bats. The book was due to be printed in June, but it's now going to come out in August. And I'm not sure if Peter Taylor is on this, is listening today or not, but Peter Taylor in the blue shirt on the left there, I would like to personally thank him for everything he's done over the years to support me and to train me up to be hopefully at least a passable taxonomist. But certainly Peter is the master, having described more bats, African bats, than anybody else alive. So let's hope Peter continues remaining healthy and alive. And then lastly, I'd like to thank the bats themselves that make my life such a pleasure. These are some photographs by Pieter Nskrecki of bats in flight. They are beautiful creatures, and let's hope that COVID does not stop us from being able to continue studying them. Sorry for the disruption, and I'm not sure if there'll be any time for questions, but thank you for listening.
Thank you very, very much, Ara. Really appreciate it, and especially with given all of the difficulties with our internet in Africa. Sorry, Peter Taylor is online, as are quite a few people who requested your talk. So I don't know if I realise that people might need to shoot off. We've got a couple of minutes, so please feel free, those of you who have another appointment and need to go, then that's absolutely fine. But for those of you who have got a little bit of time, please do feel free to stay on, and we can have a couple of minutes extra giving a talk. Ara, you've got loads of really nice messages from everybody saying how interesting and fascinating this talk was. And I'm not sure if you heard earlier, but Michelle had put a nice message on before to say that she found actually your explaining evolutionary trees very interesting rather than painful, as you had mentioned before. So
Thanks.
if any, yeah, if
Can I just say one quick thing here? Sorry, I didn't actually thank anyone in the acknowledgements, and that's because there's so many people to thank. It's impossible for me to thank. I've had a successful career of studying bats only because people have collaborated with me, and I've had a lot of very good collaborations from students all the way through to senior professors and whatever. So thanks to everybody that's worked with me. You know who you are.
Excellent. Well, thank you. And so, yeah, Peter Taylor says, thank you very much for your kind words, Ara. And Nancy Simmons said, fascinating talk, Ara. Sorry, but she had to dash off to a staff meeting. Look forward to all the publications and the book. And yeah, so we have a message from Stephanie Dippenar saying, looking forward to the update of the bats of Southern and Central Africa. So actually, how can people order that book? Can people pre-order, or where's best to get the book from?
I'm embarrassed to say I actually don't know. I've heard that you can order it on Amazon, but and I think that's fine if you're overseas, but the publisher is Wits University Press, and I think they're going to put out a call, or they're going to put out an advert or something letting Southern Africans know where to get or how to get hold of the book. And we are hoping, if COVID allows it, to have a book launch probably in the Pretoria area. So that might also be an opportunity to get a book signed by the authors. So if you live in Pretoria, Johannesburg area of South Africa, please look out for a potential book launch.
Excellent. So, and Bismarck was saying a very fascinating talk, Ara. This really highlights the challenge with identifying bats in the field and the need for lots of field work. And then Claire has a question saying, thank you, Ara. Has the manuscript for the new edition of the bats in Central Africa been finalized? Is it too late to send the notes on mistakes in the previous edition? I'm guessing. I think that means the book, not manuscript.
The book is out of our hands and with the publishers. So there is nothing we can do at this stage. But if it's very small, minor, if it's like spelling mistakes and whatever, there might be an opportunity during the final proofreading, whatever it's called, page proofs, to be able to correct those. So Claire, I'm not sure which Claire or who, but please, if you can email me, if you've got my email, please can you email me any errors that you've found? I would really greatly appreciate correcting any errors. Thank you.
Excellent, thank you. And then we had a question from Dylan saying that it's really frustrating that museum specimens appear to have mismeasured initially, or was it a relief since you confirmed the field bat species?
Well, I think this really, yes, of course, it's frustrating when something incorrect appears in the scientific literature, but we've got to remember we're dealing with humans and we all make mistakes, or whatever the case might be. The important point is that those specimens did exist and were housed in a museum that was looking after them, allowing somebody to come in and verify and check. And this really emphasizes the importance of museum collections. Without those collections, we would never be able to resolve the true identity of that Hippocydurus lamotti. So we need to value our collections and we need to invest in them, make sure that they don't deteriorate. And we also need to continue contributing to those collections in a ethically meaningful way. So without harming populations and without harming systems. We have a very long way to go. We are not going to be able to do without those museum specimens.
Excellent, thank you. And we have, so Gwen was actually saying she's already hot on the press. You can order it from their NHBS, certainly in the UK. So for anybody from Europe or North America, you might actually find there's alternative places. You can pre-order the book already, which is great. And then Aurora has asked, she said this is a very exciting indeed. So you treat Rhinolophus alticolus as an Upper Guinean endemic. Do you consider the Upper Guinean form a different subspecies from the Lower Guinean form?
I cannot answer that question, Aurora. All I know is that alticolus needs to, the whole simulator group. And in fact, if we pull back the tree that Bruce Patterson and Terry Demos, the Rhinolophus phylogeny for Africa, the simulator group is a very complex group, and alticolus is in that simulator group, at least morphologically. But I suspect that it has nothing to do with simulator, and whether the Upper and Lower Guinea forms are different or not, again, is we don't know. So we need to find out what's happening in Nigeria and compare it with what we have from Guinea.
Excellent. So Aurora says, I get you, Ara, complex indeed. And then we also have a question from Greg asking, how does the taxonomy of South African bats compare kind of worldwide taxonomy? So I guess kind of how, how comfortable are you, I guess, with the taxonomy in South African species as opposed to other countries?
Well, I think South African bats are, I'm not South African, so I shouldn't really be speaking on behalf of South Africans. But I think if you asked Peter Taylor or Lee Richards or any of the other bat people in South Africa, I think they'll tell you that we know pretty well the species identities of the bats in South Africa, and maybe not as well as Europe or North America, but not far off, I suspect. The problem is that there's lots of, I say that we know pretty well, but at the same time, there are lots of issues, and there's lots of problems, and there's lots of things that need to be resolved. And Lee Richards, I don't know if she's here or not, but she has been actively involved with some of this work herself, and hopefully she'll get to publish some of that stuff soon. As soon as you move out of South Africa, and as soon as you move out of Southern Africa, for sure, things become a lot more complicated. And there's a shorter history of collections for most of Africa. You've got to remember that most of Africa was colonized, and the specimens that were collected mostly ended up in Europe and North America. So it's very difficult for African bat biologists to be able to work in those regions without reference to those European and North American collections. So yeah, I think South Africa is probably in a little bit of a better situation than the rest of Africa.
So yeah, Lee Richards is on here, and she did say, and she couldn't agree with you more, museum collections are extremely valuable in advancing our knowledge of Afrotropical bat species. Increased access to specimen material and data thereof have never been more important. And I actually was also curious as to, because there's also been quite a lot of changes between sort of Pipistrelles and Neuromycia, and they've kind of gone back and forth. And do you think, has that changed in light of genetic advances, and it was kind of based more on morphometrics before, or why has that changed?
Yeah, so that's a complex question, but what I do promise you is that we are going to give you an answer to that question in that paper that I mentioned, that is going to be submitted this week. Part of the problem is that what appears to be similarities in morphology are in fact convergent similarities, so they're not actually similarities by descent, by heritage. And that has confused a lot of things. We've been looking at, so in other words, we've been looking at the wrong features, the wrong morphological features, and this has caused all sorts of problems. And it wasn't that anyone was at fault, it's that we didn't have genetics before.
Yeah, for sure. And I guess, there is quite a limitation, given the cost of genetics, which is obviously coming down. Yes. So I suppose, a lot of times we also, even when the technology has been there, a lot of people working in Africa, we haven't had access to labs or the funding to actually run the analysis.
Yeah.
And I was just actually wondering, it's great on the sort of batnames.org site, and it's a really great resource, but do you think there's ways of us to kind of get updates as we go, African updates on kind of as new species are described? Is there a way people can subscribe to get more information?
Well, that's a very good suggestion, and I don't see why we can't. Maybe that's something that BCA, Bat Conservation Africa, maybe could be looking into, to putting one person in charge of keeping track of what's happening on batnames.org, and then extracting the African species and presenting those to through the BCA forum, perhaps. But I agree with you, it's very difficult to keep on top of things for the world. And it's difficult enough trying to keep on top of things within Africa. Sorry, one thing I should say here, if you're not already familiar with Van Kakenberg and Seamark and Carney's now what is it called? How can I forget? The
African Chiroptera report.
African Chiroptera report. Please, it is an absolutely invaluable resource. It is, go to their site, just Google it, go to their site and download the latest version, the 2019 version. So in Africa Chiroptera report, you can have a complete list of updates, but it'll always be 1 year out of date because they publish it once a year. But those of you who are not familiar with the Africa Chiroptera report, please go and have a look at it. There is no better single resource for African bat biologists.
I mean, it really is great, and it's also they provide, it's all open source data, and they provide shapefiles and species occurrence data. It really is great. So we, as it comes out, we do publicize it, but we should probably actually do it more often than just when it's released every year. And so Helen is just saying she's really looking forward to your paper coming out. She finds Neuromysia especially very frustrating for us in Zambia. And then Ross is saying, will the new book use your new Pipistrelle phylogeny?
So the new Pipistrelle species that we're describing is from West Africa, so it's irrelevant to Southern and Central Africa. We've updated the book, but the book, I'm trying to remember when we submitted it to the publishers. It was at the beginning of this year or at the end of last year. So in other words, the book was finalized before this breakthrough that I had in Chicago at the Field Museum. So the latest developments that are coming out of Chicago, which will probably only be published later this year, in fact, probably next year, they won't be in that book in this edition. They'll have to wait for the next edition, unfortunately. I mean, I didn't know about any of these things myself until very recently.
Excellent. So I don't know if anybody has any last minute questions, but we've come to the end of it. I was just wondering, Ara, actually on a sneak peek of the new book coming out. So I believe there's some new species that are going to be in this edition.
Yeah, so we've completely updated it. Any new species that have been described, we've added. Off the top of my head, I can't remember how many that is. It's somewhere, is it 12, 15, somewhere around there. But it's not just that we've added new species, we've also updated the taxonomy that is good, at least as it was at the beginning of this year. And we've added a whole lot of new distribution records, and we've added hundreds, maybe 200 or 300 new papers, I think, that have been published in the last 10 years. So we've got lots of new photographs, lots of new bats that weren't photographed before that have got photographs now. We've got lots of new sonograms of their echolocation calls. And we've got lots of new cranial photographs. So there's a lot of holes that we've also managed to plug in. It's not just about new species, but also plugging in holes in the previous edition. This time it will be published with a cover that will actually hold, because all of you who bought the book, and I apologize for this, but the binding was not a very good job on the last edition.
Well, it was certainly a good test of who used theirs the most, I think. If you saw one that was, you know, kind of looking perfect, then, you know, that people weren't using it, and they should have been. But fantastic. Thank you very, very much, Ara. I really appreciate your time, and it's fantastic talking to you and hearing more about your work. And we're really looking forward to the book. So we'll be sure to publicise information on Bats Without Borders, but also BCA. So please keep posted of that. We'll be sharing it on Facebook and Twitter and LinkedIn. And hopefully you'll all buy the new book and support all this incredible work.
Thank you. Thanks very much, everybody. Bye.